So what are current good manufacturing practices? So they are what is in the Code of Federal Regulations that they feel makes the product safe for people to use, and make sure that if you get blood in Utah, you get blood in California, everything is still safe. Regardless of how you do it, each laboratory may do it differently, but we want the end result to be the same, a safe product for a patient. And they, again, are established to ensure the safety, quality, and purity characteristics of a drug. And those are the three words that they use a lot, the FDA, the quality, potency, and safety of a product. And they are the law, so you have to do it. You'll hear CGMP, which is current good manufacturing practices, or just GMP. A lot of people in industry just stop the C. So the C is just current. So they'll change these with any changing times, like for testing regulations, for instance. When we implemented West Nile testing, changes to hepatitis B, any of that, we're just staying current with the regular, with the technology and what's available. So why do we have to do this? Because it's a law, and it applies to everyone in transfusion service. So if you do processing, compatibility testing, if you store, if you issue anything with a blood product, then you have to follow these regulations. And that is the code that says that everyone has to. So what are the good manufacturing sections of the code that we have to follow? So you have the 210, which is current good manufacturing practices for processing, packing, and holding of drugs, because blood is a drug. So we're going to process it into its final form, and then we're going to hold it or store it until it's needed. And then 211 is the finished pharmaceuticals. So again, blood is a drug. So once it's finished, how they mostly deal with labeling and quality of the lot. So each unit of blood has a unique identifier, and that is its lot number. So in a prescription or different pharmaceuticals, there will be thousands of bottles that have the same lot number because they were all produced at the same time. So in blood products, each product that was collected from that donor at that time has the same number, and that is what they consider the lot for that particular unit. And then 606 is blood regulations. So in Part 211 is important to us because that's where it says that you must have a quality unit. And that is my position at ARUP. So you have to have someone who's an outside voice to review the things that are done in the transfusion service or the blood service laboratory. So I don't report to anyone in transfusion service. My supervisor is outside of that entire department. So I can go in and say, I really don't agree with you. And there's no chance that I could have any negative impact because that's my supervisor or she's my boss or anything like that. So you have to be removed. And the code requires that you have someone like that when you manufacture blood products. So if you're just a small transfusion service in a hospital and you're not manufacturing any products, then you wouldn't necessarily need to have a quality unit. But if you're making any products, if you're irradiating products, if you're making aliquots, you're manufacturing, so you have to have a quality unit. So if they're all three of these regulations and they're all good manufacturing practices, how do we know which ones we're supposed to follow? So the code tells us that you follow whichever is more specific, and that's also in Part 211. So if there's a regulation that applies to a situation, both in the drug section in the 200s and in the biologic section in the 600s, we're going to follow the 600 code because we're dealing with blood. So we're going to pick whichever one is more specific. And there's a lot of redundancy. So there are many cases where a situation is covered in many sections of the code, and we have to pick which one we're going to follow. And generally, you do that by whichever product you're speaking about. So the purpose, again, we want to assure the safety, purity, potency of blood products, ensure that systems involved in manufacturing are controlled and that we always get the same product at the end, to ensure patient and donor safety, and that we remain current. So again, we change things based on new diseases. So right now they're talking about chikungunya and all these new infectious agents that maybe we're going to start testing for, maybe we're not. Is there a valid test for that? What would be the impact on the blood supply? And they have a group in the FDA that reviews all these things and data and makes decisions, and then they put out guidance and new regulations if we need to follow them. So what elements are covered in good manufacturing practices? So they cover pretty much everything. So they have general provisions, they have organization, training, standard operating procedures. So they say you have to have procedures, labeling, what kind of records you have to keep, how long you have to keep them, error management, and then error reporting, which is, again, unique. They're called biologic product deviations, and we'll talk a little bit more about those, because they have to be reported to the FDA within 45 days of discovery. So what are the rules for registration versus licensure? Has everybody heard anything about blood banks being registered or licensed? Is that new? Probably. Okay, so you have to be, everyone has to be registered to have a business as a transfusion service or as a blood center. Not everyone has to be licensed. So the difference is if you are going to ship products across state lines, so they call it an interstate commerce, you have to be a licensed blood center. So to be licensed, you have to submit all of your SOPs, quality plans, submit a big form, and then all the documentation of training, how do you do training, all that type of information, and then you submit it to CBER, and then they'll come out and do what's called a pre-inspection survey, or a pre-licensure survey or inspection, and they can last anywhere from a couple of weeks to a couple of months. And these inspectors come from CBER in Maryland. So they're not the same inspectors as the people that will come, the field investigators who come and do normal inspections. So they're a little bit more focused on the back end, like how are you set up, how do you deal with changes, that type of thing. But the regulations and the ongoing inspections are always the same. But that's basically the difference between licensure and registration. So this is just a copy of what a registration or a license looks like. So you have to say, what kind of products am I collecting? So am I doing allogeneic? Am I doing autologous? What kind of things am I doing? Is it red cells? Is it frozen? Is it platelets? What am I collecting? And then what am I doing with it? Am I testing it? Am I storing it? Am I distributing it? So it basically on one sheet says everything that you do in your laboratory. Documentation is huge for the FDA. And their mantra, I guess you would say, if it wasn't documented, it didn't happen. So I could have everything except the document that says, I did this at this time and I wrote it down. You didn't do it. If you can't find the documentation for the inspector while the inspector is there, you didn't do it. You will be cited for that. So documentation is very critical for the FDA. So there are specific rules. They have to maintain concurrently, not after the fact. So if I have 6 or 7 balances that I'm doing the QC on in the morning, I can't go, this one's 500, this one's 500, this one's 500, and then write all 3 down. I have to put the weight on, write it down. Put the weight on, write it down. So it has to be concurrent. While I'm doing it, not after the fact. So like in the transfusion service, I'm reading my tubes, shaking my tubes, this is my blood type, read it, put it in. I can't read it, set it down, do something else, and then put it in the computer. It has to be concurrent as you're doing it. Everything has to be clearly traced, so each step in the process has to be clearly identified, and you have to be able to follow the process through the documentation. Has to be legible and indelible. So indelible, it can't be erased. So no gel pens. Have you ever smeared a gel pen, gotten it wet? It rubs off. No gel pens in the laboratory. No pencils, no whiteout, any of that kind of stuff. It has to identify the person performing the work. So if you're doing it in the computer, it can be your computer login. If you're doing something by hand, it needs to have your tech ID or something defined by the laboratory that shows that Becky is the one that wrote that at this time. And then it has to have dates. So I did this and put my initials in the date. So it has to show what day that you're doing stuff. It has to be detailed as necessary to have a complete history. And they don't tell you what that is. So they don't say you have to document this, this, and that, but they say you have to have a complete history. So the inspector should be able to come in, ask for a record, and be able to follow it on their own and know exactly what happened and who did it and when. And then, again, records have to be maintained and available. If you can't produce the record, it didn't happen, it wasn't done. So again, record all actions as they're happening. Use blue or black ink, no gel pens, no pencils, no whiteout or correction tape. You don't want to scratch out or write over anything. You have to be able to see what was originally written below. So if you make a mistake, everybody makes a mistake once in a while when you're writing things. You just have to use the correct procedures for your laboratory for correcting errors. Some laboratories, and generally in clinical trials situations, if you're doing testing for them, they also want to document why you made the change. It was an error. It was a typo. Why am I changing this? So that's something you may encounter as you work in laboratories. So this is just an example of really bad documentation. So we don't need any embellishments, no smiley faces, no commentary like, yes, the temperature was finally in, any of that. Write-overs, so obliterating things, arrows are not allowed unless there's a clear open and ending time for an arrow. Quotation marks are not good. So you can use an arrow if it's clear that I'm starting here and I'm ending here. But if you just have quotes, blank spots, you should never have a blank. It's the form. If you have a form and you don't need to use that space, you should fill it out as NA because I intentionally left it blank. So no oops, we don't need oops, none of that. And why are we so specific about this stuff? Because documents are legal records. So if there's ever a recall or a look back, so we find information out about a donor later on, they come back testing positive for a certain disease, we have to look back for 10 years on that donor and any donations and where that unit went to and who it went to. We need to be able to find this information and clearly understand who it is, and if it comes up in a legal case, these are going to be presented in court. They're legal documents. And they have to, again, trackability, every step from beginning to end, each critical step has to be documented. Just like the falling dominoes, this happened, and then this happened, and this happened. So, and again, if it wasn't documented, it didn't happen. So the critical information, we're documenting the who, the what, the how, and then usually there's a review. So who made sure that this information is correct? Sometimes that's in the computer system, automatically built in. Your front type has to match with your back type, or you get some sort of an error. The information you entered is not compatible with life. You'll usually get some kind of a flag in the computer system. If it's a completely manual documentation, you usually have something with peer review or supervisory review after the form is completed. And again, why do we have so many reviews? Just to make sure everything is correct and current. And the reviewer is responsible for finding errors. So we don't want to say, oops, Justin has worked in the blood bank forever. I'm reviewing his work. Let me just skim over this. It'll be fine. No, we want to make sure that you review everything thoroughly and make sure you're catching anything, because that's why we do it. If it wasn't important, we wouldn't take the time to have another person review something.